Welcome everyone to this lecture presented by myself, Dr. Muhab Al-Hinai from the Department of Biology. Today we are going to speak about the structure of nucleic acids. We are going to talk about DNA and RNA. Today we are going to talk about the central dogma of molecular biology. Basically, how do you get from a DNA molecule to an RNA molecule via the process of transcription, and then how do you get from an RNA molecule to a protein molecule via the process of translation. This is what we call the central dogma of molecular biology. And in today's lecture, we are going to talk about the structure of the DNA and RNA molecule, or what we call the nucleic acids collectively. The amino acid sequence of a protein or a polypeptide chain is programmed by a gene. Now, this gene can consist of a region of DNA, which is basically a polymer of these nucleic acids. Now, DNA and their genes is passed on to offspring by the mechanism of inheritance. So, nucleic acids store and transmit this hereditary information. Basically, as I said, there are two types of DNA molecules. We have the RNA, or what we refer to as the ribonucleic acid, and we have the DNA, or what is called the deoxyribonucleic acid. And if you look at the figure, you can see that one main difference between a DNA and RNA molecule is that RNA is single-stranded, whereas DNA is double-stranded. DNA basically provides direction for its own replication. It also directs the synthesis of an RNA molecule. RNA then controls the synthesis of a protein molecule. So how is DNA inherited from an organism, from parents to an offspring? Basically, we all get our DNA molecules from our parents, half the set from our mother's side and the other half from the father's side. Each DNA molecule is very long and usually consists of hundreds or thousands of genes. When a cell reproduces, it basically needs to make sure that it copies its DNA and gives an identical copy to each offspring or each daughter cell. Now, the DNA itself does not directly involve the day-to-day operation of the cell. You can think of the DNA as a blueprint. It basically, through the actions of RNA and proteins, it controls the actions of the cells, but it is the proteins that are responsible for implementing the instructions contained in the DNA. So you can think of the DNA as the blueprint, and the proteins are the engineers or the workers that carry out the DNA's instruction. Now, each gene along the DNA molecule directs synthesis of a specific type of messenger RNA. This mRNA then, in the case of eukaryotic organisms, leaves the nucleus and goes to the cytoplasm, where it interacts with the DNA synthesizing machinery to order the amino acids in a particular polypeptide or a particular protein. So, the flow of genetic material in a cell is basically DNA followed by RNA or mRNA, then followed by proteins. And if you look at the figure on the right, you can see that the DNA in eukaryotic cells is located inside of the nucleus. Now, when a DNA wants to express a certain gene, it synthesizes an mRNA molecule through the process of transcription. This mRNA molecule then leaves the nucleus, exits through the nuclear pores, goes to the cytoplasm, where it interacts with certain structures that we call ribosomes. The ribosomes, in the process of translation, basically translate the genetic message from the DNA into a polypeptide chain, which is basically the sequence of amino acids in a particular protein. So then, what is the structure of these nucleic acids? What are they made of? What is this DNA made of? Basically, they are made from monomers that we call nucleotides. And each nucleotide, if you look at the figure on the right, has three main components. It has a nitrogen base, a pentose sugar, and a phosphate group. And these, what we call the nucleotides. nucleotides. Now, how many kinds of nucleotides are available for the cell to make these nucleic acids? So there are two kinds of nitrogen bases. The first kind is the pyrimidines. And basically, if you look at the figure, these have a single 6-member ring. And there are three different pyrimidines. We have the cytosine, we have the thiamine, and we have the uracil. Now, in the case of an RNA molecule, uracil replaces thiamine. Next, we have the second type, which are the purines, and these differ in their structure or the structure of the nitrogen base. Basically, they have a 6-membered ring, but also this is joined by a 5-membered ring, as you can see from the figure. There are two kinds of purines available in the cell. This is the adenine and the guanine. We have then the ribose sugar. Now, this is basically a pentose, pentose meaning it has 5 carbons, and this is joined to the nitrogen bases. In the case of RNA, it is ribose, and in the case of DNA, it is deoxyribose. And from the name, you can see that the difference is that the lack of an oxygen at carbon number 2. And this is also illustrated in the figure at the bottom of the slide. Now, if these bases are missing a phosphate group, we call them nucleoside. However, if they have all three components, the pentose, the nitrogen base, as well as the phosphate, we call them as nucleotides. So how are these polynucleotides made? How are they synthesized? Basically, you have a sugar of one nucleotide that then joins a phosphate of the adjacent nucleotide by what we call a phosphodiester link. This is basically a condensation or dehydration reaction, and as you see from the figure on the right, the nitrogen base is always sticking out from this phosphate sugar backbone, and it is the sequence of nitrogen bases along the DNA molecule that give the mRNA or the gene its unique features or unique sequence. What are genes made of? Basically, genes are hundreds or sometimes thousands of nucleotides long, and the number of possible combinations of the 4 DNA bases is limitless. You have adenine, you have thiamine, you have cytosine, and you have guanine. So you can think of this as the letters of the alphabet. Depending on how you arrange the order, you can get many possible combinations of these nucleotide sequences in a gene. The linear order of these nitrogen bases in a gene then specifies or directs what we call the primary structure of a protein. The primary structure of a protein, which is basically the sequence of amino acids, then directs the 3-dimensional conformation of the protein, which in turn direct its function. Now, the inheritance of this DNA from parents to offspring depends upon the replication of the DNA double helix. The DNA double helix was first discovered in 1953 by 2 scientists that are called Watson and Crick. And unlike the RNA molecule, which is a single polynucleotide chain, the DNA double helix is 2 complementary strands. So what is the DNA double helix then? Basically, the sugar phosphate backbones of the 2 polynucleotides are on the outside of the helix, as you can see from the figure. Then the nitrogen bases basically stick out from the sides of these DNA molecules or DNA strands. And these nitrogen bases then connect to each other on the complementary strand by what we call hydrogen bonds. It is also worth noting that these DNA molecules are anti-parallel to each other. That is, they run in opposite directions. The base pairing of the DNA is basically adenine always pairs with thiamine via 2 hydrogen bonds, whereas cytosine pairs with guanine via 3 hydrogen bonds. We call the 2 strands of DNA complementary strands. Basically, what this means is if you know the sequence of one strand, you can know the sequence of the other strand, because adenine will always pair with thiamine and cytosine will always pair with guanine. Now, the transmission of DNA, basically the cell before it divides, it needs to make an identical copy of its DNA molecule. This is to ensure that one copy goes to each of the two newly formed daughter cells. So you will have a parent cell, DNA will replicate, then these cells will divide, and as you see in the figure, you will have two daughter cells with the same set of chromosomes and the identical sets of genes and DNA nucleotides. This mechanism basically ensures that the genetic information is transmitted whenever a cell reproduces, and the genetic information is passed from a parent cell to an offspring. So in this lecture, we have discussed the structure of the nucleotides or the nucleic acids, the DNA and the RNA. Next time, we are going to talk about the structures of the proteins, so make sure you stay tuned.